Well, thanks again for coming today. This is the first inaugural eHail Symposium. I'm really excited about all of you being here, and it's just really, I'm really thankful that you took your time on a Friday to come and be with us today. So my name is Akbar Walji. I'm a professor of internal medicine here at the University of Michigan. Also have an appointment in the Department of Learning and Health Sciences, and have a big interest in how to use artificial intelligence to drive equitable and efficient research in health, especially in low resource settings. So I have some work in Africa. I do a lot of work in the VA, but I am one of the conveners for eHail. Radha Mihalsi, who I'm not sure where she is, but she's one of my co-leads for eHail, along with Jenna Weens, Brahmaji Nalamuthu, and Jizu, who unfortunately couldn't be here today. But again, welcome. We were not sure how many people would attend, but we got some RSVPs from a whole bunch of folks in engineering, a whole bunch of folks from Michigan Medicine, and folks around campus, including LSA, dentistry. And again, this is exactly why we exist, and it's to bring folks like you together and the great minds to really push the boundaries of the use of technology to advance health. And so thanks again for being here today. So eHail, again, our mission is to make the University of Michigan a premier hub for eHealth and AI to improve health using technology. This was actually a joint venture between Michigan Medicine and Engineering, which started off as a sort of a strategic meeting in 2020, where we're really trying to say to ourselves, how can we prepare for the future of health using technology? And again, our colleagues in the College of Engineering, Dr. Mickelson, and our dean in Michigan Medicine, Dr. Kunkel, really were thoughtful about having us engaged and looking for gaps in where we could actually fill to bring folks across campus together. So we launched in May of 2021. And at that time, the goal was really to fill 3 different domains and gaps: community building, shared resources, and help with grant writing. I don't think we take enough advantage of the grants around NSF and NIH and really doing this co-PI model, a multi-PI model to advance, not only the science, but also the excitement around, the different projects. So eHail, again, connects and supports AI and health experts. But we don't live in a vacuum. We recognize that there's so many different entities across campus that do this work. So even though eHail is partly a union between Michigan Medicine and the College of Engineering, we recognize that there's so many other entities, either specifically in a specific disease domain or in different areas of interest around methodological advancements, not only within Michigan Medicine and not only within the College of Engineering, but across campus. Now, if anybody's missing a circle here, please come and talk to me, because again, I want to make sure that we know how we can best network with you and your group. Okay, so frequently we get asked, how are you different from this group and how are you different from that group? But really what I want to stress here is that we're trying to develop this co-PI, co-multi-PI model to push the boundaries around not only advancing the methodological expertise, but pushing the boundaries of medicine. One common example that I'm actually borrowing off Dr. Nalamuthu is, we're still struggling with what to do with one blood pressure. Now we're going to get a tsunami of blood pressures because of all the digital devices. And what is the signal? What's the noise? And how actually do we do something with that information? And that's more that, primarily would be driven by sort of advancing the methodological expertise, bringing folks like you together, and then applying for large grants that will allow us to do this work. So again, eHealth connects and supports our AI and health experts around multi-PI grants. We do that by really developing a community like this today. Grant writing expertise. We just hired a grant writer expert that could help you write your grants and really bridge that communication gap that sometimes can exist even with our own experts. And then shared resources. These are really around pilot project funding. If there's annotation that needs for large data sets, we're all preparing for these things, but we'd also love to hear from you what your needs are so we can really, again, make a difference. We have actually quite a successful, year and a half so far. We've had these co-PI model funding grants be quite successful. We've had $16 million in funding driven by some of these collaborations. But I just want to provide you one example here from Dr. Weens and her collaborators, Dr. Schoding in Michigan Medicine, really exemplifying how we would push these boundaries. So in this particular case, they're really developing AI tools around the diagnosis of acute shortness of breath, or acute dyspnea. This was funded by NHLBI in September of 2021 for $3.7 million. I know I'm not going to do enough justice to this amazing project, but Jenna, please, Jenna will share that with you if you're interested in it. But again, this is the kind of an exemplar of what we're trying to do. Again, this is our small team, but a really passionate team. I really want to thank, Henrique Florespash for really, truly helping organize this event today and making it a success. I mentioned we have a new grant writer, Hassam. I don't know where you are, but you could stand up and, oh, there he might be back there. So Hassam Abuzar is our grant writer and is at your service. So please let us know how we can be most helpful. I won't take too much more time. This is your agenda for today. We have a keynote speaker, two keynote speakers that you'll be hearing from, one this morning and one later this afternoon. We're not going to overtax you with too much stuff. There's lots of coffee breaks at times for you to intermingle and talk to each other. We have some unconference sessions that were primarily driven by your interests at 11:30. Please take the time to look at some of the posters from our students across the room. Some really, really great stuff that I think will help motivate you and energize you. And then we'll have two breakout sessions, one around successful AI health collaborations and another one around, how do we position ourselves to be really good at applying for both NSF grants and NIH grants. There's actually a new funding mechanism. Remind me again, Henrique. ARPA-H, the Advanced Research Project Agency for Health, ARPA-H, that I think they just announced a new director through the White House this week. And this is around high-risk, high-reward projects pushing against the boundaries of, again, technology and health. So, we'll tell you more about that. So please, please, please take advantage of those sessions. And I'll really kind of stop there and introduce Geoffrey Siwo. Dr. Siwo, originally born in Kenya, my homeland too. Dr. Siwo did his bachelor's degree at Egerton University in Nairobi, Kenya. Actually, really successfully navigated through a low middle income country using cyber cafes to kind of get his work done. Ultimately ended up coming to Notre Dame to do his PhD, took some time off to work at IBM, and then eventually came back to academics, and we're really lucky enough to have recruited him recently in April to Michigan Medicine. He's going to tell you a little bit about his work, and again, Geoffrey, thank you for being here and welcome.
Well, thank you, Akbar, for the very nice introduction. It's really great to be here. It's fantastic to be part of eHail, and I really enjoyed being part of the University of Michigan in the past 3 months. So I think we're living at an exciting moment. Human history has gone through different revolutions. I think we're living in an age of biology and computing. And the way I think about biology is that biology actually is also about information, that the history of information begins with biology. And biology has been here for the past 3.5 billion years. But first, I want to make some disclosures given my roles in some biotech companies as well as some other kinds of technology companies. And we know that in the past few decades, there has been tremendous advances in computing. So the speed of computing has been doubling for the past few decades. Whether or not we're reaching the end of Moore's law is debatable. But on the other side also, we've been having tremendous advances in the biological sciences. If you look at genome biology, so the discovery of DNA, the structure of DNA in the 1950s began the field of molecular biology. But it wasn't until the early 2000s when we had a breakthrough in genome science with the sequencing of the human genome. And you can see also that there are parallels between the exponential growth of computing and the exponential growth of genome sciences. For me, really my story at the interface of computing and biology begins in 2002 when I was just an undergraduate student in Egerton University in Kenya. But also had a very unique opportunity as an undergraduate student to work at this amazing research institute in Nairobi, Kenya, located within a hugely forested region. In fact, we would be having our lunch with monkeys jumping around. And this is a primate research facility that studies primates in captivity as well as in the wild. The interesting thing though at this time is that I was working with a professor who was trying to understand the role of virus-like elements in the human genome, especially in pregnancy. So amazingly, 10% of our genome consists of virus-like elements that look like retroviruses. Only 5% of our genomes contains genes. So we spent the past few decades studying only the 5% of our genomes. The 10% that looks like retroviruses is still part of the so-called dark genome. I became hugely interested in the dark genome, especially because the idea that our own genome contains retroviruses really intrigued me, because this is also a time that the AIDS pandemic was growing rapidly. So I became interested in the question, could the retrovirus-like elements in our own genomes somehow interact at the level of information, exchange information with HIV, which is a retrovirus? And I shared this idea with my professor, and he said, it's a wonderful idea, but we cannot do it, we don't have the money. But I somehow thought, what if I could explore this idea on a computer? This was in the 2000s. I didn't even own a computer then. Of course, many of us didn't even have cell phones. I think I would only seen one cell phone. But I thought, what if I could go to a cyber cafe, which is basically a place you can go and pay by the hour. We used to pay like $1, which was 60 Kenyan shillings per hour to use the internet. And I went on the internet to explore these open data sets. So the draft human genome, of course, was released in the early 2000s. So I was able to go into the human genome and begin exploring the retrovirus-like sequences in our genome. And at the information level, meaning at the sequence level, compare how the sequences are similar to those in HIV reverse transcriptase, an important drug target. And the short story is that I was able to test an idea that actually, thanks to Anthony Fauci, I came to the US in 2002 to present this idea at a conference. And just randomly, I emailed Anthony Fauci because I knew that he was the director of NIAID, the AIDS Institute at NIH. And I told him about this idea and asked him, could you support me to come to this conference? And surprisingly, I received airline tickets to come to the US. But the reason I really share this story, that this is a story about computing and the power of computing and empowering anyone in the world to achieve tremendous discoveries than they could with the resources they might need. But of course, at the heart of computing is knowledge representation. So we need to find ways of presenting our problems into data. So this has informed my research interests in 3 areas. So one in fundamental biology, especially in understanding RNA, then also in drug discovery and how AI and data could be used to advance global health. When you think about artificial intelligence, really, there's been a huge focus, of course, on machine learning and deep learning. But know that artificial intelligence goes beyond machine learning and deep learning. So things like planning problems cannot be easily tackled by deep learning. They are reasoning problems. How do you reason over knowledge knowledge which might be factual, which might not be easily represented as data. And of course, at the heart of all computing problems is how do you represent your knowledge, or how do you convert your problems into data? So in one way, it's unfortunate that AI has mostly been data-driven today. And I think this is unfortunate because actually, if you begin to think about data, there are limits of data, no matter how big. And I think the first important thing that all of us know intuitively is that, of course, data captures only a very tiny component of any physical or mental problems. And most of our problems occur in complex contexts. The other thing is that, of course, data, no matter how big again, is always going to be an instance of a problem. And the past and the future are very distinct. The past, which is data is the past, cannot be a reflection of the future, even in its totality. And most importantly, that whatever data we have to understand that the data itself is sort of a model. So it's a model in the sense that it's not the real problem, but it's a representation of the problem. So there's some loss of information when you convert a problem into data. The other thing we have to keep in mind that in fact, there are also limits of prediction, especially prediction posed in terms of machine learning. One is that when you train prediction models using data, it means that your machine learning models are by default bounded by the outcomes observed in your data. So if you're modeling two diseases, for instance, you want to know whether patients have disease A or disease B, we know that there are in fact other alternatives disease Bs that are not considered by your model. And when a patient shows up in the real world, they could be suffering from disease A, or they could be suffering from tons of other diseases that were not in your model. And of course, as AI advances into the real world, we begin to see some of the challenges of using data-driven AI. An example here is with some of the Tesla crashes, where the Tesla vehicles cannot differentiate between a moving emergency vehicle, especially if the emergency vehicle has a siren that is rotating. And this confuses the AI because the AI, when it's trained, when vision systems are trained, mostly they're trained with data that comes in frames. And when you process moving data that is moving like in a siren, it becomes very challenging for the AI to differentiate whether the object is static or whether it is moving. And of course, we can think of how those problems could emerge in healthcare also. So how do we really address these limits of data and prediction? And I think at the heart of it, of course, is that most of the problems we are solving, they are human problems. And so we have to find ways of augmenting our data with expert human insights. So this could be domain experts, for instance. We also have to think of ways to synergize AI with scientific laws and principles. Of course, recent advances in deep learning have shown that you could learn, for instance, Newton's laws of motion from tons of data. But there are many other laws or scientific principles that we know of, including in biology, that we need to design our AI systems in a way that they're consistent with those scientific principles of motion or light or biology. And importantly also, there are other ways to describe behavior or to model data. And this can include generative models, which unlike discriminative models, are not trying to classify patterns in data, but instead to generate new instances of data. And generation is interesting in that generation can allow you to explore new spaces of data that may not be available in your training data. And of course, explainable AI is very integral for the eventual success of AI in the real world. So my own research has focused on generative models, particularly has employed in de novo drug design. And particularly in one project we call the preemptive project, which started at the University of Notre Dame, because I just moved to Michigan in the past 3 months. And our goal is basically to leverage generative models for molecular design. The traditional way of drug discovery has mostly relied on screening naturally available molecules. In generative molecular design, we try to apply generative models to explore new chemical spaces by training AI models on existing chemical space. And so the goals of the preemptive project, which was in part stimulated by the SARS-CoV-2 pandemic, is basically to design broadly acting antiviral drugs. And importantly, we leverage fundamental biology to identify the pathways or processes that cells use as first-line defense against viruses. And then we try to mimic these pathways using molecules generated by AI. So just to give you a very high-level highlight of this project is that we started out with a fundamental biological problem. So in my prior research, we observed that the exposure of human cells to synthetic RNA, including CRISPR, triggers antiviral defense systems in cells. And then using a very large database of gene expression responses of human cells to thousands of drugs, we realized that actually the effect of RNA on human cells is very similar to the effect of some drugs on human cells. And so we thought that maybe we could use this idea basically to come up with small molecules that can trigger the antiviral defense system that already exists in human cells. In fact, we know that this antiviral defense system is the reason why humans don't suffer disease from most viruses, because we have multiple viruses in our environment all the time, but we rarely fall sick. Of course, this is even true for SARS-CoV-2, where not all exposures lead to infection. So then we use generative models, and I'll give a little detail on how we do this. And in addition to that, we're also interested in getting expert feedback. So either through opening these molecules to the public for feedback, as well as doing laboratory validation by testing the generated molecules on human cell lines. So we've benchmarked 5 generative models that are already in literature. So these generative models are of different architectures, including transformer models, as well as variational auto encoders. And our initial benchmarking task was basically focused on the problem on, can we use an AI model to generate nucleoside analogs? So nucleoside analogs are the most successful class of antiviral drugs. Remdesivir is a nucleoside analog. And nucleosides, of course, are components of RNA and DNA. So A, G, C, T are all nucleosides. So basically, we're interested in whether we can train an AI model that gives us other chemical structures that could mimic A, G, C, T. We also developed a new transformer model that is based on the GPT architecture. So we adopted that architecture and modified it to take in also chemical information. So information on molecular structure encoded using Morgan fingerprints. And Morgan fingerprints are commonly used in chemistry to encode molecular information in small molecules. And then we applied our AI model. So we trained it on a library of 365,000 small molecules. After training this AI model, the idea is to come up with an AI model that at least has some capability of randomly generating chemically valid molecules. And then from that, we can fine tune that AI model and focus it into a very specific molecular space. So the molecular space we are interested in is the space of AG, CET, and GO. And then of course, because of the SARS-CoV-2 pandemic, we also focused the AI model on remdesivir molnupiravir, which recently got approval from the FDA for SARS-CoV-2 infections. So just to show a summary of the results, we see that all the AI models at least are able to generate chemically valid molecules. So valid in the sense that if you look at the structure, it makes chemical sense, all the bond rules are followed and so forth. And importantly, we see that our model CRT, at least using one of the robust measures for chemical similarity, seems to perform much better than the existing models. When we look at the chemical structures generated by the AI models, we actually see that they make sense. So we see, for instance, that one of the chemical structures generated is actually similar to a molecule that we know has activity against herpes simplex virus. We also see, interestingly, that one of the AI generated molecules is actually identical to a chemical structure that was first isolated from a meteorite. And this chemical structure is similar to the A in DNA or RNA and has been found in the genomes of some bacteriophages or viruses that infect bacteria. And of course, we're interested in generating antiviral molecules. So it's very interesting that we also see that at least some of the molecules generated are very similar to naturally occurring molecules in the human body that stimulate the antiviral defense systems in our body to fight viruses like SARS-CoV-2. In fact, recent studies have shown that one of the pathways that makes a chemical structure similar to this one is very critical for natural immunity against SARS-CoV-2. So showing that perhaps targeting this pathway could be a way to target viruses like SARS-CoV-2. So we are currently trying to validate these molecules, especially the novel ones, and see whether they could actually be active against SARS-CoV-2 and other viruses. The other way of addressing the limits of data. So the first segment was about generative models, which allows us to explore new outcomes, or in our case, new chemical structures. On the other side, I think we also need to think about how to leverage fundamental biology when analyzing EHR data, because we know EHR data are becoming widely available. And in the past 2 decades, we've seen tremendous progress in genome biology. So it started with the sequencing of the human genome in 2001. We've seen the mapping of the human proteome. We've now seen the molecular profiling of human cell types. And also we have seen the profiling of human cells in disease states. So how can we use all of this data to benefit patients? Because in spite of the fact that high throughput biology has enabled us to make fundamental discoveries, the impact in the clinic has been minimal, in part because these technologies are very costly. So in contrast to high throughput biology, it's more than 5 decades since routine laboratory tests have been performed. So these tests include the complete blood count, as well as liver function tests, which are widely available. So how do we connect these two? On one side, high throughput biology, and on the other side, routinely available lab tests. Interestingly, and something that I've really learned from my mentor, Akbar, routine lab tests are also very powerful when it comes to building predictive machine learning models of disease states. Akbar, for instance, has shown that you can leverage this routine lab tests to predict risks to hepatitis, to chronic hepatitis C infections. And recently, it has also been shown that for COVID-19, you could also leverage these routine laboratory tests to predict these complex diseases. So inside the promise, the limitations of predictive models based on routine blood tests is that having a predictive model does not necessarily give you mechanistic insights on why is the model actually able to predict the disease. And I think that is an important question, because if you don't understand why, then we can't interpolate conditions under which the model might not work. The other thing is that, of course, having mechanistic insights can give us a window to develop new intervention tools for particular diseases. The other thing is that training predictive models also is very dependent on the patient cohorts that you use. So if there are any biases in the data, then these biases get carried onto your predictive models. And of course, it's very important to understand that even patients with similar disease risks may not have the same underlying biology of the disease. And in that sense, they may need different treatment strategies. So can we use fundamental biology to bridge these two worlds, high throughput biology on the one hand and EHR data on the other? And I think we can, because actually we know that there's only a finite set of genes in the human genome. So the human genome contains 10,000 protein coding genes. We will say 10,000 to 20,000. It's very hard to know the exact number. But anyway, from this finite set of genes, there are millions of possible phenotypes that can be encoded by the genome. And what basically that means is that there's no single gene for any single phenotype. So if you take any particular phenotype, be it your risk to, say, diabetes, there's not going to be a single gene that determines that. It's going to be multiple genes. So because of this, it means that when we measure any kind of phenotype, there's always going to be correlations between phenotypes. And understanding the correlations between these phenotypes can help us to walk back to what is the genetic basis of disease. So our key hypothesis is that there might be some hidden connections between some of the routinely measured analytes in the laboratory and important biomarkers for disease, especially for cancer. And just to give an example, we've been exploring this idea with publicly available protein-protein interaction data sets. So for instance here, this is a network of physical interactions between routinely measured lab analytes. So GOT1 is the gene encoding the AST liver enzyme that is routinely measured in laboratory tests. And basically we see that AST is actually connected at the molecular level to several important genes in cancer. So especially the gene EGFR, or epidermal growth factor receptor, which is a very important oncogene in several tumors. We also find that actually if we look across thousands of single cells from human liver tissue from different patients, that EGFR is actually highly co-expressed with AST across several tissues, suggesting that there's some coupling at the molecular level between AST and EGFR. And EGFR is also an important receptor for the hepatitis C virus, which is associated with increased risk to certain liver cancers. So I think the implications of this is that we should start thinking of ways in which we can bridge the gap between the high throughput biology which is very exciting for precision medicine, and what is routinely available in laboratories. And I think this is one possible way of making precision medicine more equitable by finding ways in which we can learn from high throughput data sets and apply it to low dimensional data sets that are available at the clinic. I want to say that really where we want to move into the future is the idea of incorporating fundamental biology into AI architectures. So how can we improve the design of AI models like deep neural networks with insights from biology, so that these deep neural networks, they don't have to explore a large space. We can inject in them our understanding of how genes and phenotypes are connected. And I think we can actually design a new deep neural architectures that mimic the architecture of how genes are connected to phenotypes. So I think in summary, the most important thing is to always have at the back of our minds that there are limitations of data. And if there's any way we can bring our domain expertise into the problem, that could be a way of making AI models more powerful. Another approach could be using generative models, but I think even more promising is that we need to find a way of integrating the things we know about science, fundamental science, and the things that we can observe in data. And with that, I want to say thank you very much. I'm looking forward to the rest of this meeting. I already had some fantastic conversations with a few people, and I'm looking forward to more. And again, I'm especially thankful to Dr. Akbar Walji and Henrique. Thank you for organizing this event, and to my other collaborators. Thank you.
So Geoffrey, thanks for a wonderful talk. It's interesting to see the universal nature of the cyber cafe and medical research. I think Songbird across the street gets a lot of business from us over here. So, I had a question about, the drug discovery piece is really quite interesting. You're looking for effects. Have you looked for like offsite effects for these drugs and kind of standard issues around how they might affect other biological systems? Because I'm wondering what you think next with these agents that you're able to create. Like, how do you move forward towards kind of testing? And have you done that yet in any of these examples?
Yeah. So that's a very good question. So in terms of the real world validation, so I'm collaborating with other external labs. So the Walter Reed Army Institute of Research will be performing some in vitro validation across several cell lines. So if we get some hits, then we can proceed these drugs to molecules to the next stage. Then internally, I'm also in conversations to find ways of testing some of these molecules. So obviously, off-target effects are always on our minds for side effects. And in a way, it's why we selected this specific antiviral defense pathway, because we believe that we might constrain some of the side effects of other antiviral molecules.
Right. That was a really nice talk. So I'm wondering how we can unite some of the stuff that you do with some of the more people at the medical school. Because, I learned a bunch of stuff today that I never knew, and I'm excited about the potential for finding new molecules for different things. I'm just wondering in the group, how you believe, because you're new here, what would you like to see to be able to increase the interactions between your lab, the information that you're able to generate, or the techniques that you have with, schmoes like me who are sitting in my lab trying to do things. So how do we actually do this, and how can we help you find the right partners? I think that's what this whole thing should be about, right?
Yeah, that is a good question, and one that I'm definitely learning each day. Yeah, I think it's actually interesting because so Akbara and I, we come from very different perspectives. So I am very fundamental biology driven, and I think it's very exciting that Akbar and I, we've been able to find this synergy. So I'm really looking forward to exploring how to like scale that. And I think part of it is by attending these kinds of meetings, because I already met two people actually, one from statistics and one from engineering, who had the same kind of question. Yeah, but I think there's some synergies that can definitely arise. But if anybody has other suggestions, I think this is maybe a great point to hear that.
Yeah, I do have a thought, but I don't know if how it'll be received. But one thing that we could do, and I really enjoyed your talk, and I really especially enjoyed the aspect when you're looking at drug discovery for viruses, and you were looking at the gamma or the interferon footprint. My laboratory has been studying viruses for a long time, especially the cytokine biology associated with them. And how do you align? There's a lot of investigators here that are studying inflammation. We have some very outstanding immunologists that are looking at cytokine fingerprints to identify diseases. And I think an open area that we could align research is, how do we use the immune system that's had over evolution a millennia or 3 or 4 to align itself to defend ourselves against different viruses? And is there a way to use the early response as a predictive to what sort of therapies we should be developing? And is there an AI system, kind of like I know that Akbar has done with regard to when do you use methotrexate and when do you use antibodies to TNF to start treating more advanced joint disease or IBD? So I think that it would be very interesting to align some of the other biologists here at Michigan along what you're doing in along the lines that Chuck was talking about.
In the first 30 days, I'm hoping that Geoffrey will come and meet with you. Henrique is totally following you to make sure what is it that you need in terms of resources, in terms of grant writing to pursue this further. He has every bi-weekly sessions where we'll review your specific aims. But our ultimate goal is really time focused, dedicated, sort of a targeted way of let's start these collaborations together. And we've got the time. We're doing this particularly for this reason.
Yes, that's a good point. Actually, I've been surprised in the few months I've been affiliated with Michigan, because I found that whichever area that I'm interested in, there are multiple experts in the area. And I would say particularly in, so an example is even in RNA biology, because of course nucleosides are part of RNA and are critical in synthesizing RNA vaccines or RNA therapeutics. So that's another area that it's been very exciting to know the resources at Michigan.
There was one of your slides where you were talking about crowdsourcing for feedback on the data and for the findings, and I'd be very interested in hearing more about that. I'm also interested, as part of the crowdsourcing, about a potential role for patients, caregivers, and the public as a stakeholder for, you were talking about who's left out of the pile of circles. And that would be one group that wasn't there. So,
yeah, so interesting question. Actually, yeah. In the interest of time, I had a couple of slides that were on that issue was, I think, crowdsourcing, which is basically opening problems to large communities to help you solve. That is a very important tool, especially for machine learning, because one of the challenges of machine learning is how do you objectively evaluate your model? And by objectively evaluate, I mean, if you are the modeler, you build your own machine learning model, you judge it, you say how good it is, and you publish it. How do you know that your model is the best? And this is a question that I've been interested in for several years, because because there's been studies that have shown that if you look at machine learning studies and independently evaluate them, you find that in most studies, the publishers of the paper, the performance they report is much better than the performance you get if you test that method using several metrics. So in other words, your model is only as good as the metric you use. So the studies show that if you use 5 metrics, then very few studies will be good across 5 metrics, for instance. So that's one side. The other side, of course, is that crowdsourcing allows you to also come up with new ideas in a very rapid way. And I have led one crowdsourcing challenge for malaria, where the goal was to predict drug resistance of malaria using genomics data sets. And what we saw there from over 360 participants in 31 countries is that the self-reported performance of a model is always much better than the performance you get when you use some independent data set and several metrics. And then, of course, bringing patients into having patients involved is also important. And I'm happy to discuss that on the side, because I've been both active in some consortia. So for instance, Sage Networks, which is a nonprofit organization, has been combining this both crowdsourcing from the data science side as well as participatory science in terms of engaging patients.
There's no other questions. I think we're close to our first break, but feel free again to come up to us, engage, and again, we'll be back shortly for the next sessions.
Yeah, so let me tell you a little, yeah, now you should stop clapping. So if you have a program, we're also going to put it up here. The break is self-explanatory. There is coffee over there, and as you all know, coffee goes with sweets. So there's carrot cake and other very good stuff there in the middle. I wanted to tell you a little bit about what we're going to do right after the break, and also what the system is. So the system for all of the other sessions is that the program is spatially laid out clockwise. If you're standing here, so the first on-conference sessions that are happening right after the coffee break, the first 3 are in that area, and you might want to think about what table you're going to be interested in, and then keep going clockwise. Sessions 4, 5, and 6 for the on-conference sessions that are on your program are over there. The same is for the posters. The posters are also starting there. Poster 1, 2, 3, 4. Clockwise. So that's a little bit about how the spatial layout is. And since Radha is not quite here yet, I will tell you a little bit about the unconference sessions. Those are proposed by you all, and the person who proposed them is leading the discussion. They're working sessions. We're not looking for any output from you all to give back to us, but we're hoping that if you need the supplies on the table, that you and your group make some new connections and that keep the conversation going. So no need to worry about having a rapporteur unless you want that for yourself. We will follow up with the discussion leaders and say, how can we help move this forward? You will get that email probably Monday morning at 8. Okay, coffee, sweets,